Hello, everyone. Can we have a sign of life that you can hear us? One more coming in. Hi, Lisa. Hello, Tara. Hi, Lisa. Alrighty, everyone. I know we're still a little bit early, so as people trickle in, we can let them in. But thank you for joining us today. We're super, super excited that you guys could join us, especially right before the holidays. I know that we are about to end this season with your schools and stuff. Um, Hello, Esther. But we are super excited. Just in case you haven't met me before, my name is Gemma. I'm your GBE coordinator. I'm sure I've talked to most of you. But today we are joined by Dr. Kellie Walters. She's an assistant professor of controlled environment vegetable physiology at the University of Tennessee. She's going to share some insights from her NASA-funded research about growing crops for space. She and her team test a wide range of plants, from leafy greens to culinary herbs, under LED lighting, varied harvest cycles, and controlled environments to understand yield, nutrition, and performance beyond Earth. So this is super valuable, especially as you guys are moving into the time where you guys are submitting your proposals or thinking about what to do for your second trial. This would be a great resource to ask a bunch of questions and maybe get some inspiration for trials that you are working on. I am recording this session today, so if you do have to leave early, that is okay. I'm going to put it on the GBE website after this. Otherwise, please make sure that your mic stays muted, and then at the end, we'll have time for questions. Otherwise, Miss Kelly, if you are ready, we can go ahead and begin.
Awesome. Thanks, Gemma. Let me just share my screen quick. Okay, can you guys all see my screen? Awesome. Good, good. Thank you all for being here. And I know I was originally scheduled for last week, so thanks for your flexibility. I have a new little baby boy in daycare, so we're getting all the bugs right now. But we're all doing well now. Anyway, it's really exciting to be here with you today. So I'm going to talk a little bit about what we do in my program. So I titled my little talk Controlled Environment, Controlling the Environment to Improve Yield, Nutrition, and Flavor, because that's really kind of what the overall goal of my lab is in controlled environment agriculture. So I was watching some of the old, um, getting chatting with the scientists recordings. Um, well, before we get into that, I'll talk a little bit about my background in it shortly. But one thing that I really love about controlled environment agriculture is kind of illustrated by these two pictures here. So these two pictures are of lettuce seedlings. It's one of the first experiments we did in my lab when I started here at the University of Tennessee. And it's the same cultivar. So the same cultivar, Theodore lettuce, a butterhead lettuce type. They were sown on the same day, same substrate, same watering, nutrition. The only thing that is different is the amount of light provided to these really young seedlings. And just by altering that amount of light, that light intensity, we can see very different. These plants look very different, right? The color is different. And then we do a lot of research to look into, okay, how's the nutrition different? How are other parameters different? How's the resource use efficiency? And the beauty of controlled environment agriculture is that we can control that environment to elicit these different responses. So it's really fun. Um, and that is kind of what my research revolves around. So for today's talk, I'm going to talk a little bit about my background because I saw that in these chats. It's like, how did you get to doing what you're doing? And then we'll talk a little bit about research. Now, I have a rather large lab. I have about 20 people working for me right now in my lab. And over the past 5 years, we've had, I don't know, maybe 80 people working for me. So we've done a lot of research, and I was debating, what am I going to talk about today? So first, what we'll talk about is some of our NASA-funded work. And then since our work for NASA and our work for like, the USDA and other folks here on Earth all have very similar goals. I mean, we have different targets, feeding astronauts versus feeding folks on Earth. You still want each bite you take to be tasty and nutritious, right? So very similar goals. So we'll talk about some of the work that we're doing here on Earth that I find interesting, and some collaborations that we've had with economists and kind of bringing that consumer into the fold. So again, a little bit of a different angle on things, but I thought it was interesting. And then a little bit, some other projects we're doing related to flavor and aroma of crops. So a little bit about me. I am from Northeast Iowa, small town in Northeast Iowa. Um, yeah, when I went to college, I went to Iowa State University, and I was originally going to school to be a high school agriculture teacher because my high school agriculture teacher was very influential in my life and I wanted to be like him. 'Cause he was amazing. And then I realized that you could major in horticulture and I could just work with plants all the time and not anything else. And I didn't have to teach high school to do that, even though I do love teaching in a different capacity now. And all teachers at all levels are awesome. But I realized I could just work in horticulture. So I switched my major to horticulture right before student teaching. Um, and yeah, throughout that time, I had a lot of different experiences and internships, not just throughout my undergraduate career, but throughout graduate school. So I interned at places like the Iowa Arboretum. I know there's some folks here from South Florida. I worked at Costa Farms, decided to leave the Iowa winter, which is horrible, and went to Miami for a bit. I spent some time in St. John, teaching at a private school and working in their school garden. I was in Seattle and I worked in a commercial greenhouse growing plants there. So just different experiences that helped me figure out. I pruned apple trees for a couple of Iowa winters, and maybe that's why I'm in controlled environment agriculture because I realized I hated that snow and pruning those apple trees all winter, not for me. But warm greenhouses, yes. Uh, so then I stayed at Iowa State for my master's degree. Again, working in controlled environment agriculture. I worked a lot with culinary herbs, hydroponically grown culinary herbs. That was my research. And then I went to Michigan State University for my PhD, where I did a lot more work with culinary herbs. And throughout this, I also worked with floriculture crops because I love flowers and they're beautiful. And the physiology of flowering is really cool and fun. Um, After Michigan State, I graduated in 2020 during all of that craziness, and I joined the University of Tennessee as an assistant professor of controlled environment vegetable physiology. So I mainly work with leafy greens and culinary herbs. That's where a lot of my funding is right now. But we also have projects in things like tomatoes and some other weird crops, sensor development, that type of thing too. And yeah, I'm currently an assistant professor. That just means that I don't have tenure yet, but I submitted my packet, so I should have tenure this upcoming summer. So it will be very soon. Then I will be an associate professor. So that's kind of how I got to where I am. When I was in high school, did I even think I would ever be a faculty member? No. But I did want to teach, and now I get to teach, a couple classes at the university level, and I get to individually mentor a bunch of students in my lab, which I really enjoy. So I'll be sharing some of their successes here today. So why controlled environment agriculture? You guys are all doing some plant, experiments for space application, which is really cool. But one of my goals is, okay, I took this picture in a field in the Salinas Valley in California. So the vast majority of our lettuce, for example, is grown in California and Arizona. A couple of areas that, water might not be a, a, a very, there's not a lot of rain, right? Water issues. So what happens in these kind of precarious areas that we're growing the vast majority of our produce? And if we can use controlled environment agriculture, we can diversify geographically the production and kind of fortify our food system. So initially, early on, when we were doing research, we did a big industry survey. So it's like, what areas should we work on in controlled environment agriculture? One of the questions we asked was, how much would research on manipulating the growing environment to improve crop flavor benefit your organization? And most of them said it would be moderately to very beneficial. So that's important here on Earth. It's important in space too, right? You want astronauts to actually be able to like eating their food, right? Yeah, there's always jokes about kale, right? That, oh, you don't like eating kale. But it's not just kale. It's like basil. We can drastically change how that basil tastes based on how we grow the plant. So growers here on Earth are also interested in that. And the research that we conduct on Earth and in space can be mutually beneficial. The other thing is economics, right? We asked them, will your customers pay more for crops with increased flavor? And 90% said yes. So we also have an economic case. So our overall goal is to improve crop quality, but we have to maintain yield because whether you're growing, in a greenhouse here on Earth or you're growing in space, space is expensive, space to grow plants. So you have to maintain yield and grow high quality at the same time. Because ultimately, if you're selling crops here on Earth, you're selling based on weight, right? So what is quality? Many things. So I mentioned flavor. And for our NASA work, we're also very interested in nutritional quality, right? And post-harvest longevity, particularly here on Earth. So if you harvest a crop, how long is it going to last in your refrigerator? And how you grow the crop can drastically alter how well it lasts in the refrigerator. So not something people think about as much. Also the appearance or color. If you're growing a purple head of lettuce, you actually want it to be purple, right? You don't want it to be green. You want it to be purple. Or you don't want different defects. You don't want yellowing or other weird things to happen. So quality can encompass all of these things. And also something that we're not looking into as much, but texture, right? How thick your leaves are can change based on how you grow them. Yeah. So I'm going to start talking about, again, I picked out a couple of different studies. And sometimes I talk too long, so I might have to skip things at the end if I talk too long today. But our main NASA study that we did originally was focusing on modeling leafy greens physiological and biochemical responses to light intensity and successive harvest. So I have a picture of Ethan Darby, who's the graduate student who led all of this work, did a fantastic job. Yeah. So whenever I talk about we did this, it was mainly Ethan who really did the work. So, okay, what do old long duration sea voyages, citrus, and potential missions to Mars have in common? Does anybody want to unmute and say? Potentially, yeah, Gemma?
Something to do with scurvy?
Something to do with scurvy. Yeah, vitamin C deficiency. Vitamin C deficiency can occur in about 3 months. Vitamin C degrades pretty quickly over time. And if we just have that stored space diet and that vitamin C is degrading, we don't want our astronauts getting scurvy on the way to Mars. Other nutrients are deficient in the stored space diet, like vitamin K, and other compounds can be beneficial from a human health perspective, like some of the carotenoids could potentially help mitigate, so especially male astronauts can have the swelling in the back of their eye, can help mitigate some of the swelling effects. So one solution to scurvy, we can't just have citrus lasting on that long on a minimum 21-month round trip mission, right? Without vitamin C degradation is actually growing plants to produce vitamin C and some of these other compounds that are deficient or beneficial from a human diet perspective. So that's one of our goals, is to supplement the stored space diet. Our goal isn't to provide all of the calories, it's to provide some of these nutritional components to the space diet. And from a psychological perspective, just having folks, I forgot that this had an animation that it kept moving, opening or having these are some of the compounds that we're interested in. Water-soluble vitamins like vitamin C, vitamin B1, lipid-soluble vitamins like vitamin K. Sorry, I must not have deleted some of the animation stuff. Some minerals we want to increase, and some we want to decrease, like we don't want iron. increased iron is actually not beneficial for astronauts. Carotenoids, again, can help mitigate some of that swelling. Anthocyanins are antioxidants, but they also help with that purple pigmentation. And then morphology. So we want the plant to grow quickly, but also be compact because space is a limiting factor. So we're looking at a bunch of these parameters, right? But when we're doing this, first we're building off of previous research. So I know some of you guys are familiar with Dr. Massa, Joya Massa. They evaluated a bunch of leafy green vegetable species and identified Chinese cabbage as a promising candidate. But this is what it looked like when it was grown on the space station. There were problems due to these elevated carbon dioxide concentrations. So they looked back, okay, there were further screenings and they found that Mizuna, or Brassica rapa niposynica, was a good runner-up. So we decided that to use this Mizuna as well as Brassica carinata, which we call green amara mustard greens, and some other related cultivars in our evaluations. So it's good to build off of the research of others, right? So some of the challenges and limitations in space. So I mentioned the carbon dioxide concentration, that Chinese cabbage didn't do well with that high carbon dioxide concentration. So we need to make sure that we grow crops that can handle that. There's limited space for plant production. Space is a limiting resource. Microgravity is a challenge, especially for water and nutrient delivery. And then energy partitioning for plant production. So on the space station or in further missions in space, energy is an important resource and there are a lot of competing demands for energy. So we have to keep that in mind and when we're taking our production considerations into account. So our first study, this was a picture of our first study. We did a big cultivar evaluation. So if you're evaluating different species and cultivars in your schools, this could be somewhat similar. Maybe a different production system, maybe a different scale of things. But we had, again, a bunch of different mustard greens and related species. So we grew them and we had this weighting scale. So it's like, how do you take all of these different parameters into account? So for example, fresh mass here was important. So we multiplied that by 2.5. But I mentioned before, iron is not desirable. So that was a negative weight. Then we grew the plants. And whenever we do controlled environment research, we report all of our environmental conditions so you can compare across studies. So this is what our sensors actually said, what our temperature was, what our relative humidity was. We have here bolded our carbon dioxide concentration. So we injected carbon dioxide to mimic the carbon dioxide concentrations on the space station. So we didn't have those issues like they had with the Chinese cabbage. We want to know that here on Earth, where it's less expensive to make mistakes than in space, right? And then we have our other environmental and nutritional parameters reported here. So for our, oops, I don't know why that's still circled. I guess I'm not as good at barrel drawings. This was the results from our first cultivar evaluation. Again, a cultivar is a cultivated variety. And what we found was that our top parameter was this, oops, green amara here. And it's green, right? But these other ones, they're not that much lower in our overall category. But if we want to balance some positives and negatives. So for example, green amara has high vitamin B1, which is good, but it also has high iron, which is bad, right? So we decided to also pursue another cultivar that had complementary characteristics. So we chose this hybrid red Mizuna. Now, hybrid red Mizuna had low iron, which is good, right? It balances out that green amara, but it also had low vitamin B1, which is bad. But it has anthocyanins, it has that purple or red pigmentation, which is a source of antioxidants, which is good. Also, these two crops have complementary flavors, right? So hybrid red Mizuna can be very spicy when you're eating it, versus green Amara is a much more mild flavor, almost meaty, as much as greens can be meaty. So yeah, they could be good in a mix. So it's not one size fits all. We have different crops that you can help build models for to, kind of create the nutritional profile that you desire. So then we moved on and we took these two cultivars and did a study on light intensity and photoperiod. So this was what we call a factorial design. So we had 4 different light intensities, 200, 400, 600, 800 micromoles per meter squared per second. So that's kind of a low range to a pretty high light intensity. And then we had 2 photoperiods, or 2 durations of light. So 24 hours, meaning the lights never shut off, or 16 hours. So the lights were on for 16 hours per day. And so we had 9 plants per combination that we took data on. And this was conducted twice over time. And these are what the plants looked like when we harvested them. So you can see that top row is 16-hour photoperiod, the bottom row is 24-hour photoperiod. And if you go from left to right, we're increasing that light intensity. Now, instead of showing a bunch of data, we're going to go again straight to kind of these weighted scores. And this is kind of what they looked like. Now, we were hoping that we could use a lower light intensity for a 24-hour photoperiod and have, much more efficient plants than the 16-hour photoperiod with a higher light intensity, but that really wasn't the case. So we determined that a 16-hour photoperiod was good, and we selected the top 2 kind of rankings, so that low light intensity and the highest light intensity with that 16-hour photoperiod for subsequent studies. Excuse me. So the next study that we did was on cut and come again protocols. So how does successive harvest affect phytonutrient and biomass yield over time? And we wanted to know, does allowing plants to develop more between harvests increase plant productivity over time? So the kind of theory behind this is we have, let me try to draw again. Well, we have this graph here. You sow the seed and it's kind of slow to start, right? You have cotyledons unfolding, you have to wait a bit till you get the true leaves, and they're very small for quite a while. And then you hit kind of this exponential growth phase, this linear growth phase where the plants are just putting out leaves pretty quickly. And then when plants start to reach flowering, they start to slow down. So, they're not accumulating as biomass as quickly when they reach that phase. So we want to keep them in that linear growth phase as long as possible. So that means skipping out on that propagation phase if needed. So can we grow the plant, cut off the leaves, let it regrow, cut off the leaves, let it regrow again? How many times can we do that? And we're not just interested in yield, right? That's a stress on the plant. And some of these compounds, the biosynthesis of these compounds is related to stress responses. So are we changing that nutritional profile? And so that's kind of what we're interested in. And how exactly do we do that? That was the question. So the experimental design for this was, we had kind of we separated it into 2 experiments. One was a time-based experiment. So once the plants were mature, we harvested every 10 days. So that's easy to schedule, right? Whether you're in space or here on Earth, every 10 days, you just harvest the plants. The second experiment focused on development-based harvest. So when the plant has 8 fully expanded leaves, then we harvest. Let it get 8 more leaves, then harvest again. So you have to really monitor the plant and harvest, based on development. So again, this is another factorial design. So we had 2 light intensities in that low and that highest light intensity treatment. We had 5 harvests over time. So again, here, this was the first harvest, 2, 3, 4, And then for the fifth harvest, it was only achieved with the time-based experiment, the developmental-based experiment. The plants started flowering and they leveled off and they weren't producing the leaves anymore. So I'm just going to show a little bit of data first before we get into the kind of synthesis stuff. So fresh mass, what we found with fresh mass here at these harvests were that high light intensity, we had higher fresh mass than the low light intensity, regardless of which harvest, right? This is the time-based harvest, so every 10 days, which is expected. More light, more photosynthesis, more carbon fixed, more biomass. So this is what we expected. And with that high light intensity, we started to have a drop off in mass kind of the last couple of harvests. But we're not just interested in fresh mass, right? So I pulled out a couple of parameters to show you. So beta carotene, that low light intensity, we had more inconsistencies in beta carotene concentrations than with that high light intensity. Total anthocyanins, or that purple or red pigmentation, and this was with green Amara mustard greens. We see some increases in anthocyanin concentration over time, regardless of the light intensity. And then zeaxanthin, another carotenoid, we see some inconsistencies over time and a lot of variation in the data. When we look at harvest based on plant development, so every 8 leaves, again, we see a similar trend with that high light intensity, more biomass than the low light intensity, but we have some inconsistencies in yield over time. But how do we take all of this data together to actually have meaningful recommendations? So we did a calculation on per diem output. Now, what do I mean by that? I mean, how much fresh mass is produced per day? So these dark blue dots and triangles are the high light intensity again, and light blue is low light intensity. So, and then the circles are the developmentally based harvest, so harvested every 8 leaves, versus the triangles are harvested every 10 days. And what we found was that when we're harvesting every 8 days, with that high light intensity, we have the highest fresh mass per day. Now, if we have that high light intensity and we're harvesting a little earlier because we're harvesting based on time, then we're having a similar fresh mass per day with that high light intensity as the low light intensity and that developmental based harvest queue. So again, the per day output changes quite drastically depending on what our harvest queue is and what our light intensity is. And again, we're not just interested in fresh mass, right? We're interested in some of these other parameters. And it's interesting because I pulled out 6 examples here, but regardless of which parameter, we have pretty similar results that are coming out, right? So we see towards the top from an efficiency perspective, these dark blue dots. So that high light intensity with that developmental based harvest queue was the most efficient in producing anthocyanins per day, for example, or beta carotene, or vitamin C, or vitamin B1. But I mentioned before, we're not just interested in this output per time. One of the limiting factors in production is energy. So we did these calculations for energy from lighting required, because 800 micromoles per meter squared per second light, that's a lot more light than 200 micromoles per meter squared per second, right? So when we change the perspective from per day to per unit energy, we start to see these light blue circles come out kind of on the top here. They're more efficient in producing anthocyanins or vitamin C per unit lighting energy input. So some conclusions from the NASA work that we have done, that Amara mustard greens provided kind of the best overall platform based on the weighting scale that we used, which could change if the weighting scale changes. That highest light intensity applied for a 16-hour photo period produced the best results for Amara. And when you have the consecutive harvests, efficiency is generally gained for all metrics, so it was a good thing, kind of avoiding that seedling stage every time. And the developmental based harvest, so harvesting every 8 leaves, had the greatest output. The greatest per day output was with that high light intensity, and the best per unit lighting energy is with the low light intensity. So from this study, we had a couple of undergrads that were interested in growing in the greenhouse, and we're like, okay, do some of these things apply to growers here on Earth as well? So Lauren, one of the undergrads in the lab, took some of the seedlings that were grown in this high intensity growth chamber that we were using for our NASA research and transplanted them into a common greenhouse environment, into raft hydroponic systems. So again, these treatments were just applied during the propagation phase, which in this case was about 10 days. The plants grew really quickly, and she had differences in fresh mass. So as you increase that light intensity, your seedlings increase in fresh mass. When you put them into a common greenhouse environment and grow them, again, all in the same environment, she still had really large increases in fresh mass that persisted here in this harvest graph. So from a grower perspective here on Earth, more and more growers are putting more resources into that early propagation phase because of this. Now, then another student, Maggie, went and quantified some of the phytonutrients that we were in the seedling stage and at harvest, and she saw some differences in some of the B vitamins. Just a little bit of data here, just to give you an idea of what some of the students are doing in the lab. We also had a greenhouse successive harvest experiment, and instead of focusing on developmental or temporal-based harvest cues, Sienna here wanted to look at, okay, should you harvest the basal leaves, the large leaves at the base, and leave that apical meristem, or should you cut off that top apical meristem when you're harvesting? So she was doing research related to that. And then Nathan here in the red picture was quantifying the phytonutrients and how that changed based on how we harvested the plants. Shifting gears a little bit, Gemma, do I still have time to talk about other studies, or should I? Okay, so I wanted to talk about this study because it's very simple design. So usually I design really crazy experiments. A lot of the other studies we have going on right now might be about we're building UVB LED fixtures and applying them at certain times, manipulating temperature and measuring carbon assimilation and stuff like that. But this one was really fun because we paired up with an economist, and it was focused on the state of Tennessee, but we plan to expand it across the country. And we had focused on microgreens, quick crops. So we wanted to work with brassicas. So we worked with kale and radish. And we had 2 cultivars of each, a purple leaf cultivar and a green leaf cultivar. And we had 2 light intensities. So our low and our high light. So 175 and 575 micromoles per meter squared per second. These are what the plants looked like. So one of our goals was just to have plants that look different, right? Because from a consumer perspective, it's like you're not going to know what the nutritional quality is, right? You're not going to know how it tastes unless you actually taste it, right? So we wanted them to look different. So they looked quite different. We did a bunch of nutritional quality analyses, yield, etc. Here are some of the anthocyanin data. Again, anthocyanin contributes to that purple or red pigmentation, which can contribute to this color change. And you can see that we had some differences in anthocyanins in the red cultivars and the green cultivars, differences in scale though, right? Because green has fewer anthocyanins than red leaf cultivars. And we did a lot of work with B and C vitamins as well. Because one of our goals with this was to determine Tennesseans' preferences for microgreens of a different color. So again, we had all of these colors of green here, green and purple variations with kale and radish. And we did some studies with how much they would be willing to pay. And Tennesseans, they were willing to pay more for green leaf cultivars, 52 to 66 cents more compared to light purple cultivars. And there was a price penalty for the dark purple cultivar. They did not like the dark purple cultivar. We asked them questions about which of the following techniques, like production techniques, do you perceive as being the best option to improve microgreen nutrition? And they said plant species selection, right? Which is some of what you guys are doing, but also using light to enhance the crop. And they were willing to pay 47 to 79 cents more for plants that were nutritionally enhanced via light, which was really cool. We also did some work with, okay, we quantified all of the nutritional quality, right? So we did some work with nutrition perceptions. So all the survey respondents, so we had about 900 people in this study, answered a bunch of health-related questions from things like, I consider myself health conscious, or I am constantly examining my health. And so they'd say, you know, how much they agree with this statement on. And then they, again, answered some of those questions about microgreen nutrition. So what they perceived as nutritious and their preference, you know, which ones they liked versus which they didn't. Again, they're just looking at pictures because if you're in the store, that's what you have. You're looking at the greens. And so this is a lot of data. But what I want to point out here is that they perceived this dark purple kale as being the most nutritious. So this dark purple kale, it's the deepest color that we had. And they perceived the green radish as being the least nutritious. Again, they didn't have any of our data. This is just what they perceived by looking at it. But then when they said what they would prefer after this, they said that this dark purple kale was the least preferred, even though they thought it was the most nutritious. And the green radish was the most preferred. It's kind of the opposite of what we would think. So some of our takeaways from this study is that consumers perceived that dark purple kale to be the most nutritious, but they preferred it the least. Consumers preferred that green radish despite perceiving it to have the least nutrition. And their perception of nutrition was not the main motivator in the consumer preference. So I'm trying to do research on improving the nutrition. So how do we actually communicate that with consumers and really help from a public nutrition perspective? So we did some work with those health perception questions, right? So we kind of created this health consciousness model. And our model, you know, the details aren't super important right now. But we created a health consciousness model. So maybe I'm not very health conscious, but Gemma is super health conscious, right? And then we determined what their nutrition ranking accuracy was based on our lab data for the nutritional quality of the crops. And this was derived from the most nutritious to least nutritious ranking. And we computed this correlation. And yeah, this is the graph of the accuracy. So they were not very accurate in saying, you know, what was actually nutritious. People are not good, regardless of their demographic information, their background, how much their household income is, what their educational level is, their age, none of that mattered. And everybody was equally as poor at telling which microgreens were more nutritious or less nutritious. And then, yeah, their health consciousness versus nutrition ranking accuracy, again, no correlation. Whenever you see a graph like this, it's like, okay, this is just like throwing paint at the wall. Nothing is correlated, right? So yeah, overall, we found that increasing that light intensity during microgreens production affects production by improving the yield. We're improving the leaf to stem ratio, so our quality of our crop is higher. We're improving the nutritional quality like vitamin C, total water soluble vitamins, so all the B vitamins, anthocyanin concentrations. But the light intensity also affects consumers by increasing that perceived nutritional quality, especially in red cultivars. But it negatively affects the consumer preference of kale. especially because of that coloration change. And people's health consciousness does not influence consumer ability to identify nutrition in microgreens. So we're also doing some work with this study with the Baylor College of Medicine. Baylor, Mike Dzakovic here is an assistant professor of pediatric nutrition. So we can measure, what the vitamin C is, for example, but can people actually use it? And what we found was that, of course, light can influence the bioaccessibility and how much of that vitamin C, or in this case, some of the carotenoids, can be taken up into your body. But also, very surprisingly, we found that with carotenoids, even though there's, microgreens are very nutritious and there's a lot of them in the microgreens, they're not very available to your body. Yeah, the usability is pretty low. So I know we have about 20 minutes left to the hour. Gemma, do I have some time to talk about other studies, or should I pause here?
I would say maybe another 10 or so minutes and then we'll go into questions.
That sounds good.
Perfect.
So these next studies I'm going to keep much shorter. I was just very excited about some of that microgreen data because we do a lot of plant physiology work, but one thing I love about science is collaborating with others to tell a bigger story. Like, we can grow plants to do the nutritional quality work, but we need the economists, we need the sociologists to kind of have that bigger picture together. So the next study, or the next kind of area that we're working in that I wanted to touch on, was end of production strategies. So this is a big grant that I led along with Ohio State University. We have 6 faculty working on it. Can we do something quick right at the end of production to improve the nutritional quality of crops? So we talked about how energy is important, right? Can we kind of optimize biomass, during the main portion of production and change something like light, temperature, nutrient solutions really quickly and efficiently to improve nutritional quality? So that's what our research is focusing on. Then we have an economic feasibility component, and we're putting out training resources for growers. Here are a couple of my students, Jessica and Jacob, who are working on this, and a picture of some kale in the greenhouse. And so they're in this picture, they were working on, okay, can we just increase the amount of light the plants receive right at the end of production to improve quality? And right at the end of production, what is that? Is that 1 day? Is that 3 days? Is that 7 days? So that's kind of what they're working on. And then we also had some undergraduate students, Kathleen and Evan here, working on a parallel study indoors. So for vertical farms or from a NASA application, our recommendations, should they be different in the greenhouse versus indoors? And we did have some really cool differences. This is just a graph of some of the B vitamins and how they changed based on adding that additional light. I think this was for 3 days right before harvest. But what we found in a nutshell is that indoors, it takes less additional light. So you only have to turn up the light a little bit more compared to in the greenhouse. You need to turn up the light a lot more for a longer duration to have similar responses. Now, we think that is because indoors, you can have the same light intensity all the time, right? Because you're supplying all the light, like in my office here, like if I didn't have a window, I'm supplying all the light through electrical lighting, right? Versus in a greenhouse, you're using solar radiation. And in the middle of the day, that might be much higher and then be lower later on. You can have the same photo period and the same overall amount of light or daily light integral, but you're having more variation. So the plants can adapt to that changing light intensity, versus indoors when it's the same all the time and you suddenly increase the light intensity a little bit, it's stressful for the plants, right? And some of these compounds are upregulated when the plants are stressed. So stress isn't always a bad thing. Maybe when you're taking your exams at the end of the semester, we don't want stress, but plants, a little bit of stress for plants is good. Another area that we're working on is with culinary herbs. So this is a big USDA project called CEA Herb. It's Herb Extension and Research Base. And it's focused again on marketing and economics. And then we have a big production kind of piece, that's my main area, and then engaging stakeholders, and that's a multi-university, multi-state project. And I just wanted to pick out a couple of the studies we have completed here. So this is Spencer Givens, a graduate student in the lab, and he was working on the interaction of air temperature and nutrient solution temperature in deep water culture hydroponic systems. So, okay, we're in Tennessee, and in the summertime, it gets pretty hot, and some crops don't grow well when it's really hot. So could you chill that nutrient solution that the plants are growing in, and will they be okay? Or in the wintertime, when it's cold, like it's a little chilly out right now, it's expensive to heat that greenhouse. Can you just heat that water, that nutrient solution, potentially save energy? And is that a viable option? So that's what we were working on. We were with a bunch of different crops, but I'm just showing some examples with sweet basil here. So this plant here was 6 grams. It was grown with a low nutrient solution temperature, and I'm talking in Celsius here, of about 15 degrees Celsius, an air temperature of about 20 degrees Celsius, which is about 68 Fahrenheit. So relatively low for basil. The optimum temperature for basil for the fastest biomass accumulation is about 30 degrees Celsius, sometimes up to 35 degrees Celsius. So more like 90 degrees Fahrenheit, so much warmer, right? Doesn't do as well with the cold temperature. If we just increase that nutrient solution temperature, say we're not going to heat the greenhouse, we're just going to heat that water, that nutrient solution, to about 25 degrees Celsius, we can over double that fresh mass by just heating that nutrient solution more. And if we further heat the nutrient solution kind of towards that optimal temperature, we see less fresh mass. So we were hoping we would see even more fresh mass, but it didn't work out. Our hypothesis wasn't correct. Now, if we have a higher air temperature, so more towards the optimal of about 30 degrees Celsius, and we chill that nutrient solution down to 15 degrees, our plants are about 11 grams. We have, if we don't control that nutrient solution temperature, and the nutrient solution temperature is around 25 degrees, with that high air temperature, we have about 29 grams. If we slightly heat the nutrient solution, we still have a similar fresh mass. So for basil, if the air temperature is high, we don't need to change that nutrient solution temperature. But if the air temperature is low, slightly heating the nutrient solution temperature was beneficial. Now, for other crops, our hypothesis was correct. Now, we could chill that nutrient solution slightly, especially when, during the summertime, say if you're growing a crop like cilantro, you can chill the nutrient solution a little bit and have a much higher quality crop. We also did some work with, okay, we're not just interested in yield, right? But what about some of these nutritional quality parameters? So this is just an example with flavonoids. And as we increase that air temperature, we're decreasing the total flavonoid concentration by about 58%, so pretty significantly. Now, if we just look at that nutrient solution temperature, increasing that from about 15 to about 25 degrees Celsius, we're also decreasing those concentrations by about 87%. Now, we have 2 parameters here, so if we look at them together, here we can see we have that low air temperature and low nutrient solution temperature. We have the highest total flavonoids. Well, that's the opposite of our yield results, right? So nutritional quality and yield trends are completely the opposite. So depending on what growers are interested in, they'll have to make a decision, right? But this is a culinary herb, so maybe nutritional quality isn't the highest parameter, right? We're interested in flavor, we're interested in aroma. So we quantified a bunch of the volatile compounds that contribute to flavor and aroma. And the top 15 volatile compounds here that change based on air temperature or nutrient solution temperature are listed here. Now, what does this actually mean? So 1-octanol kind of has a somewhat citrusy flavor and aroma that it contributes to basil. Transmethylcinnamate is a little bit of a strawberry-like flavor and aroma. Eugenol is clovey. Linalool, if you smell the standard, I swear it smells exactly like Fruit Loops. Every time I smell it, it smells like Fruit Loops. So all of these compounds changed based on temperature. So now that we've quantified these, our next step is to actually conduct consumer panels. So maybe all of you guys go into a room and you have a little sliding door and a basil leaf comes through and you can crush it and smell it and taste it. And then you answer questions about that, about your preference, about what you're smelling and what you're tasting. So then we can determine, okay, do consumers actually care about this change in flavor and aroma? So kind of to wrap up, I wanted to acknowledge, I only touched on a few of the studies that we conduct, but we have a lot of people in the lab who make all of this type of research go. Um, other research related to altering environmental parameters. We're doing work on developing sensors and sensor control systems for controlled environment agriculture, and it's really fun to work, collaborate with others too. And the funding for a lot of the work that we have done. And yeah, are there any questions?
Thank you, Dr. Kelly. Really quick, just before any questions come out there, if you're not able to unmute yourself, you can put it in the chat. But also, please check the chat. I've put a survey link. These surveys are really, really important for our chats with the scientists. We would really love your feedback. So that is there. If you need it later, you can also email me. But for now, feel free to unmute yourself and ask some questions. We have a few minutes.
Hi, Dr. Walters. Thank you so much for that presentation. I just have a few questions. So you said that you started measuring per diem outputs of biomass. How do you do that measurement?
Yeah, so we didn't measure them every day. So we measured at harvest and then calculated based on the amount of days that it took to get to that harvest. Yeah.
Okay.
Yeah, so
Okay.
we did not measure every day.
Okay. Thank you. Another question that maybe some of the students might also be interested in is since you do light mixtures, do you know what the light is like on Mars or the moon or in space? Like how does the intensity and the color wavelengths and everything differ in space?
Yeah, so light is really complex, and the light provided in space will change based on, okay, you can't just grow a plant in space without any kind of protection, right? So it's kind of like if growing in a greenhouse on Earth here. Different greenhouse glazings, whether it's glass, whether it's polycarbonate, whether it's acrylic, will let different amounts of light through and different light qualities through. So even here on Earth, there's a lot of research going on with spectral shifting films to alter the quality of light and the distribution of light coming to the plant, whether it's direct or diffuse. So I think from a space perspective, whether it's on Mars, whether it's on the moon, it will depend if something transparent or translucent is used to actually emit solar radiation, or whether we're providing all of that light via electrical lighting. And via electrical lighting, what I showed here is a lot of light intensity, kind of white appearing light research. But there's a lot of research going on about light quality, and with LEDs becoming less and less expensive, we can really manipulate the quality of light that we're providing to the plant. So for example, we have some research, my PhD student, Sam, he's working on, okay, can you grow the plant with white light? Maybe that's the most efficient. And then can we use a very little bit of UVB radiation, maybe even 1 minute supplied to the plant to improve the nutritional quality. So that was kind of a long answer, but if it's probably likely that we will be for a while at least just using electrical lighting where we can really manipulate the quality of light provided to the plant.
And I guess that makes also sense from your indoor versus greenhouse experiment where you did get better when it was indoor and fully controlled rather than in a greenhouse where you also had the ambient. So that makes sense.
Yeah. But from an Earth perspective, greenhouse production is much more economically viable than indoor production because energy from lighting is really expensive. So yeah, but there's some different, cost benefit analyses, right? And if you have a really intensive system, so it might make sense for microgreens, or it might make sense for really young plants when you're concentrating those resources when the plants are small and close together. So yeah, it's an interesting perspective, but we can also take those lessons learned to space too, right? So if we're trying to decide when should we use energy, when's the most efficient time to increase the light intensity, we can take those lessons into the different contexts.
I have a very silly question for you that maybe you can answer, maybe not. But when working with pink Mizunas, did you ever have any that formed a bulb?
Did we have any that formed a bulb of the pink Mizunas? I don't recall. Ethan may be able to answer that more. He did most of the hands-on work. But I know some of them had thicker stems. Um, and we didn't grow them very long. So our crop time was pretty quick. I can't remember how long exactly. Our standard for greens, we usually never go from seed to harvest longer than 5 weeks. And I know these were a lot quicker than that. I think less than 4 weeks from sowing to harvest. Besides the successive harvest experiment. But yeah, the experiments were pretty quick, so you might not have seen them that either.
Okay, thank you. We did 49 days and all of them formed like perfectly little round bulbs. I was like, I don't think you're supposed to do that.
Oh, that's cool. Hey, Lisa.
Hello, Dr. Waters, thanks so much for your presentation today. I just have a very quick question about the stress. You mentioned that at the end of the trial, you tried to put more lights on the indoor growing plants on the CEA environment. I was curious about, does that change the flavor of the plants? And I think astronaut likes to have more they taste bud was kind of like messed up because of the fluid in the microgravity environment. So I was wondering maybe that's a good strategy that we can use to alter the taste at the end of the harvest.
Yeah. So I don't have specific data on that right now from a taste perspective. We do have a lot of data on just light intensity and culinary herbs, and consumer sensory preference. I didn't show any of that, but the 200 micromoles per meter squared per second versus 600 micromoles, 1 or 200 micromoles, some of it you could put in a salad and eat basil all day, and because it was not flavorful at all. But 600 micromoles, it was so strong, it was not good. And for cilantro, I do not like cilantro. I have the gene that you taste like soap. Oh my gosh, 600 micromoles was horrible, just horrible. But 100 micromoles, I could tolerate that. That could be okay. But what one of my PhD students, Sam, is working on right now is working with UVB lighting at the end of production, so one day before harvest. And can you supply that light? We're working some of our works with light intensity, some of it's with light quality, but he's really hoping that, okay, you can have like maybe one diode and just have a little bit of light. UVB light working with it is dangerous, right? It could be carcinogenic. But he's trying to figure out a very energy efficient, time efficient strategy right at the end of production. So we'll have a bunch of data on culinary herbs here soon with the lab analyses. And in the spring, we're doing sensory analyses with it as well. But we don't have the data on that. But I think it is very promising. Yeah, but nutritional quality, it works. And we have some specific production recommendations that we'll be putting out for, greenhouse growers here on Earth for several species of leafy greens. And soon we will have more data on culinary herbs and actionable strategies as well.
Wow, that's so cool. Thank you so much.
Yeah. Yeah, that's something if you're doing experiments, is fun to test. that taste. And I can tell you with some of these greens like Mizuna, the low light intensity versus the high light intensity. Low light intensity, is okay, it's palatable. The highest light intensity, some of the students would not eat those greens because they were so spicy. They're like, this is horrible. It's way too much. But other people really like the spiciness. That's when you add that human component and like that preference and that type of thing. I think it's really fun. And I teach a plant physiology class, and I have all the students do experiments in the class. And sometimes they add that consumer perception component, and they just have random students that are going past tasting the greens, or students in the class, they solicit them and they're like, okay, what can you taste the difference in these? And it could be a cool component to add to different studies if you're proposing things. I don't know if that's feasible, Gemma, but anyway, it's just fun to taste what you're growing too.
We can't tell the students to eat the plants because they're not our students, but that is up to individual classroom teachers. With that, I think we'll call it for now. This was a fantastic talk. I personally loved it. I hope all the students and teachers that joined us today loved it as well. If anyone has any other further questions, feel free to drop them in the chat. Otherwise, thank you, Dr. Walters. This was great. This was fantastic.
Thank you so much for having me and for everybody for coming today. And yeah, have a wonderful holiday break.
You as well. All right. Bye,
Bye.
everyone. Take care.